Welcome back to our protein-protein interaction lectures here. We're going to start out with split promoters. This is an advancement over the two hybrid system, and this uses the formation of light, and that formation is very sensitive. So before we get into the other, just notice that the production is light here, and it's called the Gal screen system. So there's a galactose enzyme, and the substrate eventually forms fluorescent light, and so it's very sensitive. You can do that on a X-ray film or a fluorescent imaging that the computer imager will find and quantify. So this is super sensitive. Remember, this can be done with extracts and not necessarily inside the cell. It can be inside the cell, it doesn't have to be. Okay, so the yeast 2 hybrid was inside the cell. Okay, so that's a difference here. So this is protein-protein interactions here. This is called beta-galactosidase mutant complementation. So probably from genetics, you have heard the word complementation. Complementation implies two things are brought together to create the whole. Well, when we talked about the Gal4 transcription factor in yeast, the DNA binding in the activation domain, so you kind of split the, cleave the transcription factor, and then you fuse the bait and prey to it in the yeast 2 hybrid. That was a concept where you're breaking apart a protein and you're still able to get functioning of the domains. Well, it turns out that, and I don't know, probably not in micro, probably not in genetics, they talked about enzymes, certain enzymes can actually be broken apart, and then when they are put next to each other, they bind together and recreate the functional enzymes. Kind of a seems almost magical here. You have two pieces and two pieces of Lego, and you put them together and and I'm going to use the word magically fit, but that's just chemical bonds coming together. And so based on that concept, they wanted something more sensitive to look at protein-protein interaction. So in this cartoon here, you've got the delta alpha and the delta, and I don't know what that looks like, a W to me. So those are the two parts of the beta-galactosidase enzyme, and they're split. And in this cartoon, the bait and the prey do not interact. Notice there's no synergy, no interaction. And so you end up with no functional enzyme. And so when you add a substrate here, there's no enzyme to chop the substrate up, and there's no light. So no light there, no light. So this is a negative reaction. So you could find two proteins from the yeast 2 hybrid. You could test it this way as well. Here is the prey and the bait do come together. A does handshake with the protein B, and that as they bond together, so they recreate, they complement then the beta-galactosidase. It's now a functional enzyme. So this is the functional beta-galactosidase enzyme. When you put the substrate in, it breaks it down and releases light in a certain wavelength that is detected, and that light is a reporter. It reports to us that A and B must have found each other. That's the only way light would have come in. Certainly controls would be A by itself, B by itself, neither, right? Because you don't want A to magically uh, uh, have the capability of chewing the substrate here. Imagine A had beta galactosidase activity, or B did, and they didn't need each other, they would still give you a light. I mean, that would be very rare, but you've got to have the controls. So it's always important to have controls in your experiments, or you can't believe the data. So here's our controls. Okay. So think about controls when you would be describing this for our little quizzy poo coming up. So that's mutant complementation. And not only did they find that alpha beta galactosidase was able to be complemented, but they actually found the firefly luciferase enzyme, LUC here, would also undergo complementation. And so this cartoon shows the N terminus and the C terminus of the luciferase, and this just shows how they're put together. And basically, if X and Y are active, then you get a functional firefly gene that, under the right conditions, will give you the yellow firefly color here. So this is called, these have been called split reporters, because each protein has one part of the reporter, and when put together then by protein interaction, then the reporters are made whole again, and you can measure that, right? And so what's interesting is certainly you can test, do they interact, but you can also, once you find they interact, you can do drug inhibitors or other kinds of compounds to shut it down. Maybe there's a virus that has a protein that blocks these two interactions, and that allows the virus to be infectious. So you could test that, right? So you could also test if there's some patients, some disease where they have a mutation and the Y can't bind to X. Can you mutate the human mutant Y back so that it does interact? So it's the gene therapy concept. Can we correct what's in nature and restore the activity? So this would be a good assay system. But that's why the word is the split reporter concept, the complementation concept. So this is a little bit further here where they actually, and it's again even more sensitive, and they discuss it in the words, but long story short, they actually fuse X and Y together with a protein known as DNA-E. DNA-E is a protein that actually will splice other proteins, and kind of behaves as a protease, but in a different way. We called it splicing. And so when you read the typical central dogma that DNA is spliced, sometimes proteins are spliced. And so DNA-E is an example that can splice that. So now what we have is we have the N and the C terminus of the firefly together as well. So we have a double reporter concept here. And when X and Y bind each other, so here's what we're testing. The DNA-E is made whole, and it splices the firefly, and that's released. Okay. And then you can measure that. So you actually recreate it and put it back together, and that's a little more sensitive, because now it's not just they're next to each other. They're actually, we kind of took some tape and put them back together, molecular tape, right? And so this allows them to actually be spliced, two parts of the protein spliced together. So this is going to give you a higher, more efficient signal in your split promoter here. Needless to say, the cost of something like this is more costly than the beta-galactosidase, which is pretty cheap. So, but this is going to be more sensitive, and you're going to get a higher signal. You're saying, why do we worry? Imagine X and Y are very rare proteins. And so when they find each other, you don't get a signal and say, oh, they don't bind. No, they're just rare. The signal's just below your detection. So anytime we're doing any kind of experiment, you need to understand the parameters in science, right? And if you're going to be a doctor, pharmacist, or vet, when you give a drug treatment, what's the range? Are there super insensitive, super sensitive people? You need to understand the entire landscape, you know, as you are diagnosing. I understand when you're dealing with the average, that's most people, and you probably won't see the extremes, but the extremes are where something bad can happen, right? Oh, I missed that. So this is Dr. Bush telling you, you've got to always think, okay, that didn't work. Well, why didn't it? Is it just I'm not being able to detect it? How good is my equipment? How good is my laboratory test? How good is my detection system for that virus or bacteria? Okay, so it's not just, and again, these are the labs, and the labs, you got people that are med techs and they've got 50 samples to do per day. They've kind of got their boss saying, turn out the experiments. So you're just getting what they say, and they're doing 50 a day, and they're missing the rare situation. So you, as a doctor, need to be, or the vet or the pharmacist need to be aware of some pitfalls. In other words, where is the situation where I could be wrong? And there's a lot of thinking about thinking that I urge you to learn on your own. You won't probably get a lot of it, how to think about thinking, and in biology and certainly in medical school, but it's out there. Okay, so they can get off my soapbox here. So this is a little more information here. I found this. Here's the luciferase gene, and here it is secreted. So they've got some cells. They put this reporter in, and each one of those cells, there's a well. Here's the well here, and they add the substrate, and the luminometer is the machine that's measuring the luciferase. So these split reporters are releasing luciferase, and you're getting a light detected. So for looking at 96 different interactions here, you can scan your luminometer, actually exposed kind of as a bingo card, A1, A2, A3, and you get a reading. Okay, you get a reading in the computer, and you can then go back and see what was in that well. Okay, so it's called a luciferase, and the machine is called a luminometer. And you can put these reporters in. Here's a living cell, here's luciferase, and you can actually do real-time monitoring, watching luciferase turn on and off. And here's the luminometer, and you can watch over time the intensity. And if there's some process, you can watch, oh, it's high and then it drops. Is it, so is that S phase in the cell cycle? Is that mitosis? Is that G1? What is that, G2? So you can actually do some real-time reporting, and this is more in molecular biology. We'll talk more about real-time reporting, but you've at least seen it here. And then here's the plate concept, where you incubate for 10 to 60 minutes. I'm not sure the procedure, but anyway, you're detecting light, and you send your luminometer and detect through each of the little bingo cards here, and you're getting detection. Here's the Lu and N-terminus and C-terminus, and this is showing how the luciferase, and you add the substrate, it's called luciferin, and here is the production of light. See, they show a sun here, and here they are, luciferase and the X and Y are found together, and just showing the process here. And this is just a reporter gene for transcription. Okay, and that can be a reporter for transcription. We talked a little bit about DNA binding and activation, and here's the light produced right here. And so this is a reporter. So I just wanted you to realize there's a machine and there's a chemical reaction, a substrate, and we're measuring downstream of the actual interaction. Okay, so that's called a reporter. And here's the complementation, the split luciferase. And so I show you an experiment where they actually took plasmids that are PC part of the luciferase, PD, PI. These are only parts of the luciferase. And they mixed PC with PD and they got perfect complementation. The PD with the PI got no interaction, and the control is the complete luciferase, and looks like cells were harvested with these possibilities. And the way we measure on the lunometer is called RLUs. These are light units per microgram of protein. So you're measuring everything in these split reporter assays based on the amount of protein. So for instance, if you run in and say, oh, I got no light units produced, they don't work. And I say, well, how much protein was in there? And you didn't do it. How do I know that you didn't put any extract? You forgot the telephone, your iPhone rang and you forgot, or you put 1/100th of what you should have. And so you always measure against the amount of protein, total protein in your extracts. And that's a control, right? How much protein is there? So now you look at light units versus the amount of protein. And so here's the content, here's the PK. So this is the positive control where they put the complete gene in. Here's the negative control, they put no gene in. And here's the various PC by itself, PD, PI, and then they mixed them together and they got complementation only with these two parts of the luciferase, not here. Okay, so that just shows you this is relative light units per microgram of protein. So the complementation is about 1/2, right? About 50% of the full. And you're saying that doesn't sound too good. Well, you can get a signal. This is pretty high anyway. It's not like it's so low. If something was close to 0, that would be an issue, but this is high enough we can detect it. So this is probably good enough for an assay. And somebody else did another experiment where they cut it to different ways and did the same thing when they added things together. And it was a reconstitution assay, and they found these regions of the luciferase would complement here. So people have been taking different parts and finding the most efficient split reporter. So everything I've showed you at this point, this has been done, and they know this is optimized for the best reporting using the split reporters here. And there's some more data on the experiment here. And just to confirm, when they did a Western blot, here's the complete intact protein. Look at this. So even though they were split, if you allowed them to find each other, they recreated and behaved as a complete protein, even though they were made in two parts. So you don't hear a lot about this in genetics. Classically, before they knew this, if you said, hey, if I broke an enzyme apart, you would say, oh, the enzyme won't work. That's not true. Okay, so sometimes enzymes can work if you complement the two parts. Depends on the parts, right? Parts is parts. And then here's showing some of the time course where you can actually measure interactions over time. And this shows the data here from 0, 18, and 36 hours. And the mock is cells that did not have any luciferase put into them. You can't even detect luciferase relative light units there. And then you see the complementation at 18 and 36 hours here. Okay, so that was looking at soluble proteins. What about membrane proteins? Uh, we will find in some of the later work that membrane proteins can bind to each other either very stably, transiently, that binding can be influenced by signal transduction, whereas it can have two membrane proteins dimerize or come together based on a fusion with a hormone or some type of growth factor. And so they wanted to develop a protein-protein interaction to measure membrane protein interactions if two proteins were in a membrane. And so what they invented, and you'll love the term here, they called it cub and nub. So there you go, cub and nub. And in this so-called dual membrane system, the reporter protein is a fusion of the DNA binding domain of the LEX-A transcription. That's a transcription factor, TF. Okay, and the herpes simplex virus VP16 transactivator or activation domain. So remember in the yeast 2 hybrid, the GAL4 had a binding domain and an activation domain. Guess what? That is very common. And the LEX-A has a binding domain and is an essential, a human-based virus carries around an activator that turns on and off our genes. They carry the keys to the kingdom here. Not good. We don't want a herpes virus to turn on our genes or turn them off. But yet they have proteins that in fact just do that. So when I teach virology again, we can go into some more detail about, and VP means viral protein. It's 16 kilodalton. That's where that number. So it's a viral protein, 16 kilodaltons. Remember, each 330 dalton is an amino acid, so you can figure out how many amino acids that is in a 16 kilodalton there. So it'll be 1600 divided, no, 16,000 divided by 330, something like that. Yeah. So the reporter, this hybrid reporter, LEX-A herpes VP16, is fused to the cub moiety, which is fused to an integral membrane, and it's called the cub TF, and that's sitting in a membrane. And then any prey we want will be fused to a transmembrane protein that has the nub moiety to it. So what does that look like? Well, here is the cub moiety. Here's the membrane protein. The cub has been fused to it, and also the VP16 LEX-A has been fused to it. So it's this a three-way or three different proteins or parts of proteins fused together here. And the concept is that if there's a cut here, a protease splicing here, that releases this transcription factor, this kind of hybrid transcription factor. If there's one there, then the cub could come along, and that may or may not be what we want. The nub looks like this, and it sits right there. And here is the nub. So you can see, does this or that interact with the nub? That's kind of where we're going. So the only requirement for the dual system is cub and nub must be on the same side of the membrane. And the interaction shouldn't lead to uh, that does not lead to the splitting off of the transcription factor will result in cells that are His minus or lac minus. So if it's His minus, they'll die. If it's lac minus, they won't have a blue color. And if the bait and prey do interact, cub and nub are brought together where they will form a enzyme known as ubiquitin, split ubiquitin, which will actually is a protein cleavage. It's a protease that will cleave that, let's say, VP16 reporter. It has a nuclear localization sequence, which you're finding about in your inquiry, and it binds and activates reporter genes such as the His, so they'll live, or the cells turn blue in a beta galactosidase. And so here's that cartoon. The cub and nub come together, and if they don't come together, then that no interaction occurs. If they do come together, we have a splicing, because this is a protein cleavage splicer. This is a ubiquinase, and it chops this, and that then finds its way through the nuclear pore, binds to a reporter, and you get a histidine or you get LAC-Z. So you live or you turn the colonies blue there. So now we can test, do protein 1 and 2, are they handshaking? Okay, this is not soluble, these are membrane. Okay, so now we have a way to do soluble, and now we have a way to test membrane proteins. There are limitations, and I think in the 2 hybrid I showed you the weakness. So just because we have an interaction by the 2 hybrid, the split promoters, or the dual membrane system, we need something else to show the interaction really takes place. And so we will pick up with that in the next recording here. So have fun, come back and see me.